let's talk Chancelloriids. So you'll remember these from before. Chancelloriids are related to Chancelloria. The id is just like turning it into an adjective. And these are sponge-like organisms, but with hollow spicules, which is weird because sponges don't have hollow spicules as far as we know. And they'll also have this sort of tuft of spines that are covering the major opening on the top. Ostia in the side and osculum in the top. And so what you'll see when they get fossilized, here's a picture under fluorescent light. Here are some sponges. There's a Chancelloriid next to them. Here is, I think, a brachiopod, and I don't know if that's a sponge, but this is a Chancelloriid for sure, growing off it. Here's a couple of Chancelloриids on their own, along with what look like a couple of brachiopods. There's a brachiopod apparently living attached to a Chancelloriid. And I think those spines are there to prevent exactly this from happening. So, you had one job, spines, and you failed. Here is Chancelloria pentacta. Imagine a space between those words, and then you got it. And here's a group of them. These are from Utah, I believe from the Margam Formation out there, where you get lots and lots of trilobites and awesome kind of middle Cambrian things. Yeah, it's a pretty cool formation as they go. Yeah. So, remember we said they are hollow spicules, and they definitely are. These hollow spicules are something of a mystery. So we don't know if they're made by a different process. Is there like a cell on the inside that's secreting structure on the outside of it? Or possibly there was some kind of organic core, like maybe a protein core, and then it's just mineralized on the outside. And if that's the case, the only difference between these and sponge spicules is that the core has not been preserved. The core was made of something else and then mineralized over the top. Yeah. Which is possible. Yeah. Like the way our bones start out as cartilage and then we mineralize those up and turn them into, hard bones. Yeah. So certainly could happen. Wrapping something around the outside of a spongier, earlier component that's easier to lay down. This is, I don't know if it's the most famous Chancellorid, but it's the one we have the most copies of. So, yeah, be looking out for this. This is Allonnia, and you can recognize Allonnia because it's got spicules that either look like this, or they'll have 3 spines and one sticking up in the middle, or they'll have 4 spines, like a front one, a back one, and 2 side ones. As opposed to Chancelloria, which is like a wagon wheel that has groups of 5 or 6 or 7 spines coming out from it. Here you can see the cluster of little spines up around the top, and we think these are probably there to keep parasites from invading. Like there's a lot of kinds of worms that would love to do no work whatsoever. Let the sponge do all the water pumping, and to go in and parasitize the sponge and eat all that food, or chew on the sponge cells themselves. All of which is disastrously bad if you're a sponge. So the idea is that these may have been sponge-like, but they're Chancelloриids, they're their own group. Yeah, but sponge equivalent, perhaps. Genetically similar. Now you find Chancellorid pieces before you find actual Chancelloриids. Here are examples of small shelly fauna. You find these from the earliest Cambrian. These are just where you get a rock and you can dissolve the rock, and there are no bodies like of complete articulated things preserved from this time for whatever reason, and that's a hotly debated point. It's probably something microbial that we can't really see, like a piece of missing information. But what you'll see are a lot of these things where you'll have a sort of a central disc and then maybe 6 or 7 little rays coming off the sides. And those are going to be hollow inside, like here's a little hollow one. And you'll have some that are flat, like a wagon wheel, just like Chancelloria. And you'll have some that are kind of angled, like, little devil horns. And that's just like Allonnia. Yeah. So yeah, and you have some that are in groups of three with one little short one and a medium one and a long one. And I don't know who that is, but probably related to Allonnia because they're all sort of pointed up like that. Then you got all these other things. The sclerotome, the range of hard parts from a variety of organisms. We actually have some idea where these come from. It's thought to be a primitive mollusk for at least some of these guys. Some other ones may be foraminifera, possibly. Some of these are little tubes that had some kind of organism inside of different tubes. And we think they're probably animal, but it's hard to say exactly what. Tomotids are one of the names of these things. I think some of these even go back to the Precambrian, if I remember right. But definitely in the Cambrian, early Cambrian, you do find lots of these small shelly fauna. All right, so the structure of a Chancelloriid is actually preserved. So all the ones we're going to see are from the Burgess Shale up in Canada. And so you remember Burgess Shale preservation, right? All the hard parts are gone, but we have casts of the hard parts. And all the soft parts are beautifully preserved. They were like coated in minerals, and then they were dissolved. And basically the minerals were washed out, but we were left with a beautiful impression that preserves the soft parts. So here you can see this texture. Look at that. That was the texture of the outside of an Allonnia right there. And here you can show that in situ on some actual Allonnia fossils. So it's like a leathery coating. Here's a bit of different preservation. You can see this, look at that. Pyrite preservation is what I think we've got going on there. So this is a pyritized, like fool's gold replaced version of a sponge. And here you can see that little, the opening, the big central opening at the top. And look at all those little spines. Look at them all growing all around there. A little different from the structure of the spines. And so if you imagine this thing dying, rotting, falling apart, getting spread out, you might find spines that look like this and spines that look like this, and they might be from the same animal but have a different appearance. So that's the thing with those small shelly fauna. You can see what's there, you can categorize them, but it's kind of hard to tell who's who unless you can find an intact body fossil from around the same time or just a little later that gives you a clue. Yeah. So very conservative interpretations. This one is Allonnia, so this guy, but Allonnia nuda, because it's basically naked. Look, there's a set of spines, empty space, there's a spine, empty space, there's a spine. It's barely got any spines on. That's no way to run a Chancelloriid organism at all, now is it? There are only a few specimens of this. It is pretty rare. But on one of those specimens, if you zoom in to the real tiny stuff, like little tiny bumps like that, you'll see that they're actually little impressions of rings, which are probably the pores that are bringing water in. These may be individual porocytes that are leaving these rings. They're a little bit larger than you would expect, so maybe it's a group of cells forming a pore. But yeah, that's the idea. And so this is the paper that actually showed that Chancelloreids are probably pulling water in at the sides and out at the top and covered in spines like a lot of sponges are. And so this is what led to them being classified as sponge grade organisms. Their own kind of thing, but very spongy and probably related to sponges. And you could kind of put them within the crown group with the sponges. Yeah, if you're the kind of person that just got to pigeonhole everything, you could do that, I guess. I don't know. So here we are. Here's the opening. There's the little spines surrounding that, along with the big spines over here in our Allonnia nuda, which is almost completely nude of spines. Poor Allonnia. Put some spines on. Oh my gosh, don't go out of the house looking like that. Here is a type of living sponge. So this is an actual extant living sponge. It's one of the Calcaria, if I remember right. And this one, you can see how much of this. So the green is staining the living body of the sponge. This is a slice through it. The black parts are spicules. So this sponge is mostly spicule. There's very little actual meat on this thing, but you can see it here. And look at this. This one has a cluster of different looking spines, and they are all around the big opening at the top. And so here's an intact one where they've still stained it up so you can see where the cells are. And so based on this, saying, hey, look, there's something very similar that's around now, and finding all these features that we didn't know were present in Chancelloriids before, we can basically say that, yeah, they're probably about as spongy as things could get in the early to middle Cambrian. So, all right, review questions. Yeah, let's wrap this up. What are Chancelloриids? They're kind of like sponges, but they have hollow spicules. And they have usually a lot of spines covering the main hole, probably to keep parasites out. They have a leathery texture on the outside. They have little pore-like things that were made of soft tissue. They didn't leave any sort of hard tissue trace there or mineralization. So yeah, that's the evidence. Describe the anatomy of a Chancelloriid. Just did. Yeah, go back and look. Go back and watch. What are small shelly fauna? Little things with hard parts. And so these are among the first organisms on the planet that figured out that they needed to protect themselves. And the question is, what were they trying to protect themselves from? Because it's easier to find the prey than the predators. Because, yeah, think of the Serengeti. Think of the plains of Namibia. Tons and tons of zebras and gnus and, food animals, basically. Very few of the big cat predators. Yeah. But it's actually the right balance, because if you have more of the big cats, then you sort of crash the prey population, and then the big cats population also crashes, and then you get back to equilibrium with a lot more little gazelles than lions anyway. So let's see. Chancelloриids are a big component of small shelly fauna, and those date from far before we have actual body specimens of organized Chancelloриids. So we know they were there, we just don't have good fossils of complete ones, which is a shame. And it may be that they exist in other places and will eventually be found, and I, for one, will think that's pretty cool when they do. What's the evidence for water flow? Pores on the side, big hole in the top. Other than that, you can't see water flowing in the fossil. You can just say, you know what, it had all the equipment, it would make sense. And they have sort of a solid leathery coating in between those pores, which probably wouldn't allow water to pass through. So this is directed water flow is what they are built for, if you like. Yeah, they built themselves, but, yeah. What are porocytes? They're the cells that make pores. We covered that already. That was last lecture. And what's the theory for the form of the main open? Like, why are they so spiky? Probably keeping out parasites. That's most likely thing. There are shrimp that live in sponges and parasitize them for life and actually get trapped inside of there and will live their entire life and die inside of there. Sometimes you get two shrimp trapped in there together. Like their little embryos will come in and they'll develop inside and live well off the sponge. But if there's a male and a female in there, they're trapped forever and they can at least breed. A lot of times, though, you get two little males in there, or two little females, and then they're just stuck there forever with each other and no more babies. Yeah, not that I think shrimp necessarily have really good relationships with each other, but there you go. Yeah, and so I think that's plenty on Chancelloриids. I think you'll agree. But those are worth knowing, Chancelloria and Allonnia. For the test, and like this information about like what are they? Yeah, how do they work? What do they do? So thanks a lot.